So, no good discussion of cells is complete without discussing germ theory of disease. And in order to go a little bit deeper into that, we're also going to discuss the immune system. And this is all part of our Unit 3 on the structure and function of cells. So, let's get into what this germ theory really is. Be able to define it, explain how we developed it, and what humans believed before the germ theory. So prior to understanding that things like genetics and cells could cause disease, humans had a lot of supernatural causes for disease. Things like imbalances in our humors, too much blood or not enough blood, too much phlegm or not enough phlegm. Things like curses or sins. God could curse you if you weren't following the proper rules, and that could make you sick. Or miasma, meaning bad air. You were just around an area that had bad air and it caused a disease in you. Now, this whole idea of supernatural causes was also tied into this idea of spontaneous generation, where life could come from nothing, and not complex life like humans. Obviously, we came from other humans, animals come from other animals, but simple life like insects, they could just kind of appear. Their eggs just kind of came from nowhere. So the first step to really understanding that disease could be caused by living organisms really started with this idea of disproving spontaneous generation. And there was a scientist in the 1600s named Redi, and he was the first to really do an experiment that helped us disprove this idea of spontaneous generation. First, what he did was took raw meat and put it in 3 different types of containers. One where it was completely sealed to the outside, another where there was just some gauze on the top of the container separating the meat from the air, and one where there was no top at all. It was just a container that was open completely. Now, people believed back then that as the meat spoiled, it produced that miasma, and that miasma would just cause maggots and flies to appear. Now, Redi believed that was not the case. The maggots had to come from somewhere. So when we do this experiment, he found that the flask that was sealed completely, where no air exchange was happening, that meat did not form any maggots as it spoiled. Now, the one that was covered with gauze, there were maggots that appeared on the surface of the gauze where the organisms, the flies, could come and land and lay their eggs, but none appeared on the meat inside because the flies could not get inside. And the one where the flies were able to get on top of the meat itself, lay their eggs, this is where the maggots then started to grow. So he said, look, the flies have to come, and these maggots come from the flies. They don't just appear out of nowhere. The flies must lay eggs, and those eggs must turn into maggots. Then we have Louis Pasteur, a couple hundred years later, kind of redid Francesco Redi's experiment and did it with bacteria. So what he did was he took a broth, which is basically the media that, like a liquid that bacteria like to live in, that has lots of good nutrients in it, and he boiled it. And by boiling it, you destroy all the living things inside. You kill all the bacteria in there. And this flask had a very special neck. It was called a swan neck. And what this allowed was air could go inside and pass into the media, into the broth, but bacteria could not. They could not get up the neck and fight against gravity to be able to get inside. And it was thin enough that most insects and things like that couldn't get in as well. So by boiling it and killing everything inside and trapping all of the microbes from the air in that swan neck, he could leave this container out for weeks at a time and nothing grew inside that nutrient-dense media, even though it's a favored food of bacteria. But then he took an open flask and repeated the same experiment, boiled it to kill everything inside, but then left it open for things to fall in from air currents and gravity. and that carried a bunch of bacteria in it, and the media became contaminated, showing that even for bacteria, they don't just come from nowhere. They have to come from another bacteria that makes its way into the media and is alive. So now Robert Koch was a surgeon who found that infectious diseases could be caused by microorganisms, by bacteria. So his professor had a hypothesis that these bacteria could cause disease, and he was the one that did all the experiments to prove that this was true. Basically, he saw a rat that had a disease, and he isolated the bacteria from that dead rat. So he took samples from the blood and the body of the rat, grew them on media, and bacteria started to grow. He then identified, okay, this is what the bacteria looks like. He made notes of what it was. Then he took some of that culture and gave it to a perfectly healthy rat. That healthy rat then got sick and died in the same exact way as that original rat did. Then he took samples from the body of that rat and looked at it after it grew, and it was the same exact organism. So he found that I pulled this organism from one animal that died, I isolated it, gave it to a new healthy animal. The animal got sick in the same exact way and died in the same exact way. And when I opened its body, here was this cell that wasn't there before. That was the same thing I found in this dead animal. We might think, no duh, in today's day and age, but someone had to discover these things first. And in this case, it just happened to be Dr. Koch. So this led to the development of what we call the germ theory of disease, that germs or bacteria can cause disease. Microorganisms can make people sick. So we call these microorganisms pathogens, harmful, disease-causing, most often bacteria. So this is not the only way that disease is spread. We've since then learned that you can get sick from genetic diseases, being injured, of course, but the contagious diseases, the ones that we can pass from one to another, are started by bacteria or viruses. So let's get into how our body responds to this introduction of a foreign object that can cause disease, like a bacteria or a virus, by learning about the immune system. So be able to identify the first, second, and third lines of defense and know what they are, and then identify the different types of immune cells and their functions. So the immune system, the whole job of this system is to respond to and eliminate foreign invaders and other dangers to the body. So this protects us against bacteria, viruses, parasites, fungi, anything that's not meant to be in our bodies. It can also protect us from contamination from other human products. So when blood that isn't ours mixes with our blood, we have a reaction to it that's driven by the immune system. It can also protect us from our own cells that have turned against us, ones that have become cancerous. They can detect our own cells that are behaving oddly because they've been infected by a virus. So it's a very fascinating body system. Now, the one thing that we really don't like about the immune system is that it unfortunately overreacts sometimes to things that we encounter day to day, like pollen. And so the immune system is also responsible for a lot of our allergies. So there's two different divisions, or what this means is we split the immune system into two parts: the innate or non-specific immune system and the adaptive or the specific immune system. The innate immune system is a bunch of structures and built-in responses and cells that already exist in our body that respond to any external threat in the cell. And we are all born with this subset of structures, responses, and cells. We don't have to learn them. We don't have to earn them. They just come built into our DNA. So this is going to include our first and second line of defense against invading organisms. Our adaptive or specific immune system is responsible for the targeted attacks against very specific invaders. So this is where we get sick with something like the flu, and then the following year, if we get exposed to that flu again, our body can remember it and fight it off better than it did before. So this is the immunity that we have to learn and develop over time. We don't just get this the second we're born. And this is going to include our third line of defense. So let's talk about the first line of defense, our first part of the innate immune system. These are just our physical barriers, things like our skin and our mucous membranes. Our mucous membranes are the kind of wet skin linings of our body. So your nasal passages, your mouth, your urethra, your anus, things like that. And these all have attributes that help make them antimicrobial, or basically make them able to fight off things like bacteria and viruses. Some of these are things like having keratinized cells. These are dead cells that make up most of our skin. And since they are dead, they're very dehydrated, they're very tough, they're very hard for cells to break through. We also secrete sebum and other things to help make the surface of our body more acidic, because a lot of organisms don't want to live in very highly acidic environments. We also do this with salt. Our sweat is incredibly salty, and that produces a very negative environment for cells to want to live on our skin. In our mucous membranes, we produce mucus. This is a sticky carbohydrate that physically traps pathogens and keeps them from being able to move deeper into our bodies. In our tears and in our saliva, we produce an enzyme called lysozyme that actively can kill bacteria. And then we have our normal flora. These are the bacteria that humans are meant to live with, the bacteria that lives on us and in us that can actually protect us from pathogens and foreign invaders that want to do us harm. We also have something called the ciliary elevator. This is a lot of cilia, or little tiny hairs inside our respiratory system, particularly our trachea, the part that leads down into our lungs. And these cilia are constantly beating upwards, and any mucus that makes their way into that area of our body, into the trachea, the ciliary elevator pushes that mucus up and back toward your throat, so you can then swallow that mucus and all the bacteria caught in it and send that to your stomach, where they can be destroyed. We also have hairs in our nasal passages that help to trap bacteria, so we can then clean that out later by blowing our nose or things like that. It prevents them from getting into our nice clean lungs. Our second line of defense is still part of the innate immune system, the part that we're born with. So these are groups of cells, chemicals, and responses that are going to protect our body should our outside barriers, our first line of defense, should that fail. This is going to include things like our inflammation response, fever, phagocytes. These are our white blood cells that go out and eat bacteria. NK, or natural killer cells, complement, and interferon. Let's get into a couple of these now. So inflammation has a very specific purpose. It's not just something that's there to make us miserable. The purpose is to prevent the spread of the pathogen and to help bring other immune cells to deal with the problem area. So this is a response that's triggered when our body's cells are either damaged or destroyed. And there's 4 signs of inflammation. Redness. That redness is there because we are bringing more blood to the area, and that tends to make the area look more red because our blood cells are red. Swelling is the next one. Once again, we're bringing more blood and more fluid to the area, so that's going to cause it to swell up. There's going to be heat because we store a lot of heat in our blood. So the more blood comes to the area with inflammation, the hotter that part of our skin is going to feel. And it brings pain because as we bring more blood to the area, getting a theme with this? As we bring that more blood to the area, that's causing more fluid to fill that area, and it's pushing on other structures as it swells up, and that is interpreted by our body as pain. So the whole point is to bring more blood to that area, and blood contains our white blood cells. Those white blood cells and other structures can then leave the blood cell or leave the blood vessels, enter the damaged area, and eat up or destroy any invading bacteria that made its way into our body. So these white blood cells, we call them phagocytes. That means cells that eat, and they're just going to go around and eat debris, eat bacteria, eat any kind of foreign invader for us. So they surround the organism or the debris and package it up into a vesicle and bring that vesicle into the body. This vesicle or package is called the phagosome. That phagosome will then fuse with a lysosome, the stomach of the cell. The lysosome then gives the phagosome all of its enzymes and acids and hydrogen peroxide, and all those things help digest the foreign invader or the bacteria, the debris, whatever it might be. So you can see in this top GIF here that the big cell right here is actually a white blood cell that is traveling around trying to get that little bacteria, those two dots that look like this, and it's trying to eat them up. And all these other cells are red blood cells. The next part of our second line of defense is fever. This is when our body intentionally changes its internal temperature. We try to increase the internal temperature in response to an infection. So our white blood cells can make these chemicals called pyrogens, and these are just chemicals that force our body temperature higher. They're little messengers that get sent up to the brain and say, hey, the white blood cells have detected an invader, make it get hotter in here so we can take them out. So the high body temperature causes a few things to happen. First, there's many viruses and bacteria that do not like high temperatures. This will cause some of their proteins to denature and they can't function as well, so they don't survive very long at high temperatures. But neither do we really, so we can't have a fever indefinitely. The fever also speeds up our body's chemical reactions, so it heals faster, it can go after bacteria more efficiently, the viruses more efficiently, things like that. And very interestingly, when we have a fever, the higher temperature signals to our livers to store more iron and zinc. These are often minerals that are hard for bacteria to find in nature, but they're very important for the function of their enzymes. So bacteria will often come looking in our bodies for those things. They'll want to get into our blood cells and break them open and get at the iron and the zinc. So by storing up all of the extra iron and zinc in our body, in our liver, it can't feed, not really feed, they don't use it for food, but it can't go into these bacteria and help them copy themselves and make the infection worse. It's kind of like starving them in a way. It's starving them of the materials that they need to make copies of themselves and to perform at optimal levels. There's also NK cells, complement, and interferon. These are all still part of our second line of defense. Natural killer cells, or NK cells, are just special cells that wander around in our blood and our lymph nodes. Our lymph is a type of fluid that filters in a separate set of vessels similar to our blood. And they go around the body looking for our own cells that are not behaving properly. Ones that have become cancerous, ones that have been infected by a virus. And then what they do is they poke holes in the membrane of that cell, causing our cell to burst and die. So it says, here's a problem cell, I'm going to poke holes in it, kill it, and then we don't have to worry about it again. Then there's complement. This is just a group of proteins that exists in the plasma, in the liquid part of our blood. And it sticks to foreign invading cells or viruses or particles, and they send out a signal saying, okay, we found something, inflammation response, start here. So that inflammation response then brings blood to the area, which brings white blood cells to the area and causes them to be able to eat whatever foreign invader has made its way in. And then there's interferon. This is a protein that is released by cells that have been infected by a virus. So it's our cell's way of throwing out a message saying, please help, a virus has attacked me. It warns all of the neighbor cells around it so that they can put into place various responses that helps protect them against viral infections. Then we have the next line, or the second part of our immune system, and our third line of defense. This is the specific or adaptive immune response. This is the one we have to learn over time. It's not just built in and happens immediately. So this is where we target specific invaders for being the flu or being COVID or something like that. And it includes our B cells and our T cells. You've probably heard of those before. So there's 3 key aspects to this specific immune response. It is specific to the infectious agent or the pathogen. It's going to affect the whole body, not just the area where cells have been damaged. And it remembers past infections. This is where the memory comes into play. That's why it's not something that happens immediately. We have to be infected by the thing first so we can remember it for the next time. So we break up our specific immunity into a couple different smaller parts. The first one is humoral immunity. We call this also antibody-mediated immunity. You've probably heard of antibodies before. They're just tiny little proteins that are made by our B cells, and they bind to this thing called an antigen. All cells have multiple different types of antigens. Antigens are just proteins or carbohydrates that exist on the cell membrane that help identify the cell. So your cells have little antigens on them that say, oh, well, I'm Amanda's cell. I'm not just Amanda's cell, I'm Amanda's liver cell. I'm Amanda's muscle cell. So we have lots of different antigens that label what the cell is. And these antibodies, they're little proteins that look like this. And they can attach to those antigens on the surface of a cell. That's really an antibody, but just kind of pretend it's an antigen. It's a signal on the top of the cell. They attach to that and they cause responses in our cell. So these B cells will bind to an antigen and that activates them. So it all starts with the B cell. It's coming around, it sees a bacteria or a virus, and it connects to the antigens on the surface of that bacteria. And then this turns the B cell on. Now the B cell is going to make a lot of different copies of itself. Some of these copies become memory B cells, and they're going to just circle around in your bloodstream waiting for that reaction or that infection to come back around again. Other ones become plasma cells, and the plasma cells make those antibodies for us, and so the antibodies then go out and will bind to that specific antigen that the B cell originally attached to. So as I said, cells can have many different types of antigens. So you can have multiple B cells attached to the same cell, and all of them will make different antibodies because they could possibly attach to different antigens. Now these antibodies will go out and target and attach to all of those other similar antigens. Then you have the T cells. T cells are sent out to perform targeted attacks on our own cells. So the B cells are producing antibodies and attaching themselves to other invaders like bacteria. The T cells go around checking on all of our own cells. If they're not behaving properly, they're going to target them. Now, they might not be behaving properly because they're, infected with a virus or they're cancerous. So there's cytotoxic T cells. Cyto means cells, so these are T cells that are toxic to cells. And they kill cells that are infected by viruses. So they can also kill cancer cells. They can also kill cells that are like, hey, you're a human cell, but you're not Amanda's human cell. You don't belong here. They'll attack those too. Then you have helper T cells. These ones go and recruit other immune cells like our white blood cells and things like that and saying, hey, we've identified an infection. You need to come help us take it out. Then we have memory T cells. These are ones kind of like our memory B cells that are just going to remain in the body, and they will respond to any future attacks by a pathogen that we've seen before. So that is it. If you have any questions, just let me know.